Welcome to the deep dive. We're here to basically pull out the key ideas from different sources, giving you a shortcut to getting up to speed. Today, we're diving into animal physiology, specifically those really intricate communication networks that will keep everything ticking along smoothly inside an animal's body. Think about something like the northern gannet. These birds, they dive into the freezing North Sea. But what's amazing is how the female keeps her eggs warm. She doesn't use a brood patch like many birds. Instead, she wraps the webbing of her feet around them, holding them at a perfect 37 degrees Celsius. It's just, well, a fantastic example of precise biological control. And this kind of control, it isn't magic, right? It's all down to communication. Constant, complex chatter within cells and across the whole body. So that's our mission today, to unpack the world of endocrine systems. We're talking hormones, those chemical messengers controlling everything from growth to metabolism to, how the body handles stress. We'll be using animal physiology, from genes to organisms, second edition, as our guide. Okay, let's get into this communication web that makes animals work, sometimes in really unexpected ways.
It's fascinating, really, when you trace this back. We think of hormones as complex, but basic cellular communication. That's ancient. It was around even before multicellular life. Even single-celled organisms, protozoans, they release chemicals, pheromones for mating, for instance. And they have internal local signals too, paracrine signals. This chemical signaling is the foundation for everything else.
So it's built on something incredibly old. Okay, but how does this endocrine system differ from, say, the nervous system?
Yeah.
They both communicate, but differently.
That's a really key distinction, the nervous system. It's built for speed, rapid, precise responses, mainly for dealing with the outside world. Think reflexes, quick muscle actions. The endocrine system, though, it's more about duration, controlling activities that need to last, coordinating different tissues like the liver, muscles, the gut, over longer timescales. A big evolutionary step was developing internal body cavities and circulatory systems. That created like a highway system for these signal molecules, the hormones, secreted by ductless glands to travel long distances. And crucially, these two systems, nervous and endocrine, they're often very closely linked. You have neurons secreting neurohormones right into body fluids. It shows that deep connection.
Okay, so it comes down to the messengers themselves. What are hormones made of chemically?
Right, they fall into three main chemical groups, and their structure is fundamental to how they work. First, you've got peptides and proteins, basically chains of amino acids. Insulin's a good example. Most animal hormones fit here. Second, the amines. These are derived from one amino acid, tyrosine. Think thyroid hormones or stress hormones like epinephrine. And third, steroids. These are lipids, fats, derived from cholesterol, like the molting hormones in insects, ecdysone, or sex hormones, estrogen and testosterone. And insects, interestingly, they can't make ecdysone from scratch. They need the precursors in their diet.
So structure is key. And I've heard solubility, whether they dissolve in water or fat, is a really big deal. Why is that?
Yeah, solubility. It dictates, well, pretty much everything. How a hormone is synthesized, how it's stored, how it's released, how it travels in the blood, and critically, how it interacts with its target cell. So hydrophilic ones, peptides, catecholamines, so they're water-soluble. Lipophilic ones, steroids. Thyroid hormones, they're fat-soluble, not very water-soluble. This difference shapes their whole journey.
Right. Okay, so how does that play out when they're actually being made and released?
Well, for peptide hormones, they're made like other proteins that get exported from the cell, often as larger inactive prohormones that get chopped up into the active form. They're then stored in little packages, vesicles, and released quickly when needed by exocytosis. Allows for a very rapid response. Steroid hormones, though, totally different. They start from cholesterol. But the key thing is they can't be stored. As soon as they're made, they just diffuse right out of the cell. This means the rate they're secreted is completely controlled by the rate they're made. Different organs have specific enzymes to produce specific steroids. Amines, the tyrosine ones, they are stored before release, kind of bridging the gap.
Storage is a major difference then. Okay, once they're out, traveling in the blood, how do they get to where they need to go?
The water-soluble ones, the peptides and catecholamines, they just dissolve and travel freely in the plasma. Easy. But the fat-soluble ones, steroids and thyroid hormones, they can't do that. They almost always bind to specific carrier proteins in the plasma. And here's a really important point. Only the tiny amount that's unbound, floating free, is actually biologically active and can leave the blood to reach target cells. The protein-bound stuff acts like a big reservoir, releasing more free hormone as needed.
And how do they actually work when they arrive? They don't just affect any old cell, do they?
No, exactly. Specificity is key. Hormones only act on target cells that have the correct receptors for them. These receptors can be on the cell surface or inside the cell. Hydrophilic hormones, the water-soluble ones, typically bind to receptors on the surface of the target cell. This binding often triggers rapid events inside the cell, maybe changing ion flow or activating what we call second messenger systems like KMP, which then alter existing proteins to cause the effect. Quick changes. Lipophilic hormones, the fat-soluble ones, they do differently. They can slip right through the cell membrane. Their receptors are usually internal, either in the cytoplasm or the nucleus. And here's the really cool part. These internal receptors are often transcription factors. So the hormone binds its receptor, and this whole complex then binds to specific parts of the DNA, called hormone response elements.
Wow, like flipping a genetic switch?
Precisely. It activates specific genes. The cell then transcribes that gene into messenger RNA, which leads to the synthesis of new proteins. And it's these new proteins that carry out the hormone's effects, often leading to longer-term changes in the cell. And evolutionarily, these internal receptors are ancient. Genomic studies show related receptors even in mollusks, like the sea slug Aplysia, having an estrogen-like receptor, suggests steroid signaling was around way before chordates. It's also worth mentioning that some steroids can have faster, non-genomic effects too, acting on membranes or enzymes, not just genes.
Incredible. A tiny molecule triggering new protein production. It really sounds like a little hormone goes a very long way.
Oh, absolutely. Hormones are potent at incredibly low concentrations. We're talking picograms per milliliter, that's a trillionth of a gram. Contrast that with neurotransmitters, which act locally at much higher concentrations. The power comes from amplification. One hormone molecule binding its receptor can set off a chain reaction, a cascade, that results in thousands or millions of final active protein products, a huge amplifying effect from a tiny initial signal.
Okay, so they're incredibly powerful. How does the body keep them in check? You can't just have them flooding the system all the time.
Right, regulation is critical. The main way is by adjusting the rate of secretion.
Mm-hmm.
And negative feedback is a huge part of this. It's a really common control mechanism. Think of the thyroid example in mammals. If thyroid hormone levels in the blood drop, the pituitary gland releases thyroid-stimulating hormone, TSH. TSH tells the thyroid gland to make more hormone. Then, as thyroid hormone levels rise, that hormone itself feeds back and inhibits the pituitary from releasing more TSH. It keeps the levels relatively stable, a self-regulating loop.
Makes sense, but sometimes you need a sudden burst, right? Not just stable levels, like in an emergency.
Exactly. That's where neuroendocrine reflexes come in. These trigger sudden increases in hormone release in response to specific stimuli, often external ones. The classic example is the adrenal medulla pumping out epinephrine and adrenaline during stress, fight or flight. Some systems actually use both. They have negative feedback for the baseline levels, but also these reflexes for sudden adjustments, like cortisol during prolonged stress.
And timing seems crucial, too. Many hormones follow daily cycles, don't they?
They do. Most hormone levels aren't constant. They fluctuate rhythmically. The most common is the diurnal or circadian rhythm, that roughly 24-hour cycle. This rhythm drives oscillations in gene expression, physiology, behavior. Cortisol, for instance, typically peaks in the morning, helps you wake up, get ready for activity. These rhythms are driven by internal biological clocks, endogenous oscillators within cells. But these internal clocks need to be synchronized with the outside world. External cues, called Zeitgebers, the light-dark cycle, is the big one and train these rhythms. It's a form of anticipatory regulation, getting the body ready for predictable changes. It raises a neat point. If almost every cell has clock genes, why have a master clock in the brain? Well, in mammals, the suprachiasmatic nucleus, the SCN, acts like the conductor of an orchestra.
Ah, coordinating all the individual players.
Exactly. It synchronizes all the different organs, each with its own internal rhythm, so they all work together in time, using special clock genes found in pretty much all eukaryotes.
So disrupting that synchronization like jet lag or shift work really throws things off, not just sleep.
Absolutely. And beyond secretion, hormone levels are also affected by how they're transported, broken down, and excreted. The liver and kidneys are key players in inactivating and removing hormones. Generally, hydrophilic hormones break down quickly, minutes to hours. Lipophilic ones, bound to those plasma proteins, stick around longer hours to maybe even a week.
And the target cells themselves, they're not just passive, are they? Can they change how they respond?
Good point. No, they're not passive. Target cell responsiveness can change. One key mechanism is downregulation. If a cell is exposed to high levels of a hormone for a long time, it can actually reduce the number of receptors it has for that hormone. We see this with insulin and type 2 diabetes related to obesity. It's kind of local negative feedback, making the cell less sensitive, a form of acclimatization, really.
And hormones can influence each other's effects too.
Yes, definitely. There are several ways. Permissiveness is when one hormone is needed for another hormone to exert its full effect. For example, thyroid hormone increases the target cell response to epinephrine. Synergism is when hormones working together have an effect greater than just adding their individual effects up. Like FSH and testosterone both being needed for normal sperm production together, they achieve more than either alone. And antagonism is when one hormone opposes the action of another. Progesterone inhibiting the uterine contracting effects of estrogen during pregnancy is a classic example. Keeps the uterus quiet.
It's a really intricate dance. And this isn't just a vertebrate thing, right? Non-vertebrates have complex systems too.
Oh, absolutely. The diversity in nonvertebrates is immense. You see endocrine control in mollusks, like the sea slugoplegia with its egg-laying hormone. Annelids, like leeches, have hormones resembling our oxytocin. Crustaceans, like crabs and lobsters, have well-defined systems. Glands controlling molting, others in the eye stalks controlling color change, blood sugar reproduction. It's quite sophisticated.
But insects seem like the real masters of hormonal control, especially with molting and metamorphosis. How does that work?
Yeah, insects are a fantastic example.
Yeah.
Because they have that hard exoskeleton, they have to shed it periodically to grow. That's ecdysis or molting. This whole process is under tight hormonal control. Two key hormones are ecdysone, a steroid that actually triggers the molt, and juvenile hormone, or JH. Ecdysone essentially tells the cells to prepare for molting and growth, acting via nuclear receptors to change gene expression. But JH is where it gets really interesting for metamorphosis. The amount of JH present determines the type of molt.
How so?
Well, if ecdysone is released when JH levels are high, the insect molts, but it stays in a larval form, just gets bigger. If ecdysone is released when JH levels are low, it molts into a pupa. And if ecdysone acts when there's essentially no JH, it molts to the adult form.
Wow. So JH is like a status quo hormone for the larval stage.
Kind of, yeah. It dictates whether larval characteristics are retained. Classic experiments prove this. If you remove the glands that make JH from a young larva, it molts prematurely into a tiny adult. If you give extra JH to an older larva that should be pupating, it just molts into another, larger larva.
That's amazing control over development. What about pheromones in insects? You mentioned them earlier, not quite hormones, but similar.
Right. Pheromones are chemical signals released outside the body that affect other individuals of the same species. So, not internal regulators like hormones, but they work via chemical signaling. Their sensitivity can be incredible. There's a famous story about Jean-Henri Fabre, a French naturalist who had a female emperor moth emerge in his lab, and within hours, dozens of males arrive, having detected her pheromones from potentially kilometers away. They're used for all sorts of things, mating, trail marking, alarms, especially in social insects like ants and bees, where they coordinate the entire colony, almost acting like hormones for the superorganism.
Okay, moving back to vertebrates. Let's talk central glands, especially the pituitary. It used to be called the master gland, but that's not quite right, is it? The hypothalamus is really in charge.
Exactly. The pituitary is critically important, but it takes its orders from the hypothalamus above it. The pituitary has two main parts, functionally distinct. The posterior pituitary isn't really a gland in the traditional sense. It's more like an extension of the hypothalamus made of nerve endings. It doesn't make hormones. It stores and releases two neurohormones that are actually synthesized up in the hypothalamus.
Which ones are those?
Vasopressin, also called ADH, or antidiuretic hormone, which helps the kidneys conserve water and also constricts blood vessels. And oxytocin, famous for its role in uterine contractions during birth and milk ejection during nursing. Vasopressin has an ancient ancestor, vasotocin, found across many vertebrates with roles in water balance and reproduction.
So the posterior part is storage and release. What about the anterior pituitary?
The anterior pituitary is a true endocrine gland. It synthesizes and secretes its own hormones. It makes 6 major ones we know well: growth hormone, GH, thyroid-stimulating hormone, TSH, adrenocorticotropic hormone, ACTH, and then 2 gonadotropins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH, plus prolactin.
And how do they work? Similar to the other hormone types.
Mostly, yes. TSH, ACTH, FSH, and LH typically use G-protein-coupled receptors linked to the CAMP second messenger system. GH and prolactin use a different pathway called the JAK-STAT pathway, which also ultimately affects gene transcription.
But how does the hypothalamus tell the anterior pituitary what to do? It's not a direct nerve connection like the posterior lobe.
Good question. It uses a special blood vessel connection, the hypothalamic hypophyseal portal system. It's basically a mini circulatory system, a capillary bed in the hypothalamus connected directly to another capillary bed in the anterior pituitary. This lets the hypothalamus release tiny amounts of its own releasing hormones and inhibiting hormones directly onto the pituitary cells without them getting diluted in the general circulation. Very efficient control.
So that sets up those hierarchical chains, like hypothalamus tells pituitary, tells thyroid.
Exactly. That's the classic three-hormone axis or hierarchy. Hypothalamus releases hormone 1, which tells the anterior pituitary to release hormone 2, like TSH, which travels to the target endocrine gland, like the thyroid, and tells it to release hormone 3, thyroid hormone. And crucially, negative feedback loops regulate this whole cascade. Hormone 3, the one from the final target gland, feeds back to inhibit both the anterior pituitary and the hypothalamus. Keeps everything in balance. It's how hormonal birth control works, using synthetic estrogen and progesterone to suppress the pituitary's release of FSH and LH, preventing ovulation.
Makes sense. What about the intermediate lobe? I know it's small in humans, but important elsewhere.
Yes, the intermediate lobe, tucked between the anterior and posterior pituitary, is prominent in many vertebrates, though rudimentary in adult humans. Its main job is secreting melanocyte-stimulating hormones, MSHs. MSH controls skin coloration by causing pigment cells, melanocytes, to disperse their melanin granules, making the skin appear darker. Vital for camouflage in amphibians, reptiles, fish. It's also involved in seasonal coat color changes in animals like snowshoe hares.
Okay, let's shift focus a bit. Growth and managing fuel. Huge jobs for the endocrine system. Growth isn't just gaining weight, is it?
No. True growth involves making more protein, cells dividing, bones getting longer. It's an increase in structural mass, distinct from just accumulating fat or water. And while growth hormone GH is obviously central, growth is incredibly complex. It needs an adequate diet. Malnutrition, especially early in life, causes stunting. Think about human brain growth in the first 2 years.
So it's not just more GH equals more growth. It's a whole orchestra.
Definitely. Freedom from chronic stress is also key. Stress hormones like cortisol actively inhibit growth. You see this effect in, say, crowded fish in fish farms. Other hormones are permissive or contribute to thyroid hormone, insulin, sex hormones. The pubertal growth spurt is a great example. It's driven by a surge in GH, plus the effects of androgens, testosterone in males, adrenal androgens in females. But eventually, these same sex hormones cause the growth plates and bones to fuse, stopping further lengthening. And much of GH's growth effect isn't direct. It stimulates the liver and other tissues to produce insulin-like growth factors, IGFs, primarily IGFI.
Cool.
These IGFs then act on the target tissues to promote growth, acting through pathways similar to insulin.
Okay, what about managing energy, the body's fuel supply?
Right, fuel metabolism. Bounding anabolism, building things up, storing energy and catabolism, breaking things down, releasing energy. Since we eat intermittently, we have to store fuel. And the main storage depot. Adipose tissue, fat. Excess carbs, fats, even protein can be converted to triglycerides and stored in fat cells. Why fat? It's incredibly energy dense and doesn't require water for storage, unlike glycogen, stored glucose, much more efficient. Think emperor penguins fasting for months on their fat reserves.
And maintaining blood glucose seems absolutely critical, especially for the brain.
Hugely critical. The brain normally relies almost exclusively on glucose. So blood glucose is kept in a pretty narrow range, about 70 to 110 milligrams per deciliter in humans. If you fast for a while, the body protects the brain supply. Other tissues start burning more fat, glucose-sparing, and the liver ramps up gluconeogenesis, making new glucose from amino acids. There are some amazing adaptations like the crucian carp surviving months without oxygen, partly by packing its brain with glycogen and drastically lowering energy use. But too much glucose, hyperglycemia, is also bad, dehydrates cells, damages proteins.
So who are the main hormonal players managing blood glucose?
That would be the pancreatic hormones. Insulin and glucagon, secreted by clusters of cells in the pancreas called the islets of Langerhans. Beta cells make insulin, alpha cells make glucagon. Insulin is the hormone of plenty, or feasting. Its main job is to lower blood glucose, plus amino acids and fatty acids, by promoting their uptake and storage. It tells cells, like muscle and fat, to take up glucose by inserting special transporters, GLUT4, into their membranes. Insulin secretion is primarily stimulated by a rise in blood glucose after a meal classic negative feedback. Amino acids and certain gut hormones also stimulate it, anticipating the nutrient influx. Glucagon, on the other hand, is the hormone of fasting. It generally opposes insulin. Its main effect is to raise blood glucose by stimulating the liver to break down stored glycogen, glycogenolysis, and make new glucose, gluconeogenesis. It also promotes fat breakdown. Glucagon's main stimulus is a fall in blood glucose. Again, negative feedback.
So they're like a push-pull system, insulin lowering glucose, glucagon raising it, keeping things balanced whether you've just eaten or are fasting.
Exactly. A beautiful antagonistic pairing. Interestingly, a high-protein, low-carb meal actually stimulates both. Insulin responds to the amino acids, but glucagon also responds, and its glucose-raising effect counteracts insulin's potential to cause hypoglycemia from the protein stimulation alone. Keeps glucose stable. Other hormones like epinephrine, cortisol, GH, and thyroid hormone also affect metabolism, generally tending to raise blood glucose and fatty acids. They're often called insulin antagonists or diabetogenic hormones.
And when insulin signaling fails, that leads to diabetes mellitus.
Correct. Diabetes is essentially characterized by the metabolic state of prolonged fasting, even when the person has eaten. Inadequate insulin action, either lack of it, type 1, or cells not responding properly, type 2 insulin resistance, combined with inappropriately high glucagon, leads to chronic hyperglycemia and other metabolic problems.
Okay, one last major area, calcium. You said it's incredibly tightly controlled. Why is calcium so critical?
Calcium ions, Ca2+, are fundamental to so many processes that plasma levels are kept remarkably constant. Even small deviations have immediate serious effects. Number one is neuromuscular excitability. Low plasma calcium makes nerve and muscle membranes overly excitable, leading to spasms, even potentially fatal asphyxiation if respiratory muscles are affected. High calcium depresses excitability, causing sluggishness, cardiac issues. It all relates to how calcium affects sodium channel permeability. It's also vital for excitation, contraction, coupling, and muscle, especially cardiac and smooth muscle. And for stimulus secretion, coupling the release of neurotransmitters and hormones relies on calcium influx, plus roles in cell junctions, blood clotting. It's everywhere.
Wow, so how does the body manage it, especially when, as you said, most of it is locked up in bone?
It involves constant exchange between the extracellular fluid, like blood plasma, and three main compartments: bone, the kidneys, and the intestine. Short-term, minute-to-minute homeostasis is managed mainly by rapid exchanges with bone and adjusting how much calcium is lost in urine. Long-term balance, maintaining total body calcium, depends on matching intestinal absorption to urinary excretion. Three key hormones orchestrate this: parathyroid hormone, PTH, calcitonin, and vitamin D.
Let's start with PTH. Sounds important.
PTH is the principal regulator. It's secreted by the tiny parathyroid glands, usually 4 of them nestled near the thyroid. Its main effect is to increase free plasma Ka2+ levels. It also lowers plasma phosphate. PTH is absolutely essential for life. Without it, calcium drops too low.
How does it raise calcium? Does it just dissolve bone?
It acts on bone, kidneys, and indirectly on the intestine. Bone is constantly being remodeled, built by osteoblasts, broken down by osteoclasts. PTH influences this balance. It has fast effects that stimulate specialized bone cells, osteocytes, and osteoblasts to pump calcium rapidly from the fluid within bone canals into the plasma. This is like a quick withdrawal from the bone bank account without actually breaking down the structure. It also has a slower chronic effect. It increases the activity of osteoclasts, the bone-dissolving cells, which releases both calcium and phosphate from the mineralized bone matrix itself. So prolonged high PTH does lead to bone loss. In the kidneys, PTH increases calcium reabsorption, saving it from urine, and increases phosphate excretion. And critically, it stimulates the kidneys to activate vitamin D.
Okay, what about calcitonin? Is it just the opposite of PTH?
Calcitonin is secreted by C cells in the thyroid gland in mammals. It does lower plasma calcium, mainly by inhibiting osteoclast activity, reducing bone breakdown. But here's the interesting thing. In mammals, its role in routine day-to-day calcium regulation seems surprisingly minor compared to PTH. Removal of the thyroid doesn't usually cause major issues with calcium control as long as the parathyroids are intact. It might be more important during periods of high calcium stress, like pregnancy or lactation, protecting the skeleton from excessive loss. In some fish, though, it's much more potent.
And vitamin D. You said it's actually a hormone, not just a vitamin we get from food.
That's right. Vitamin D can be obtained from diet, but it's also synthesized in the skin from a cholesterol derivative using sunlight, UVB radiation. So, technically, the skin acts as an endocrine gland, producing a precursor hormone. This precursor is inactive. It needs 2 chemical modifications, first in the liver, then in the kidneys, to become the fully active hormone, calcitriol or 1025-dihydroxy vitamin D3. And PTH is what stimulates that final, crucial activation step in the kidney. Active vitamin D's main job is to dramatically increase calcium absorption from the food in the intestine. It does this by boosting the production of calcium-binding proteins and transporters in the gut lining. It also helps PTH act on bone.
So what happens when this calcium regulation goes wrong?
You can get various problems. PTH hypersecretion, Too much PTH, maybe from a tumor or chronic low calcium intake, leads to hypercalcemia, high blood calcium. Symptoms include muscle weakness, kidney stones, neurological issues, and bone thinning. The old mnemonic is bones, stones, abdominal groans, and psychic moans. Vitamin D deficiency means poor calcium absorption. PTH levels rise to compensate, pulling calcium from bones. This leads to soft, demineralized bones, rickets in children, bowed legs, osteomalacia in adults. You also see problems during periods of extreme calcium demand. Parturient paresis, or milk fever, in high-producing dairy cows is a classic example. Just after calving, the massive calcium drain into milk can overwhelm the cow's ability to mobilize calcium quickly enough, causing severe hypocalcemia and collapse.
That sounds dramatic.
It is. Interestingly, managing their diet before calving, sometimes by feeding lower calcium, can actually help prevent it by priming the PTH system to be more responsive when the demand hits. Egg-laying birds face similar challenges, putting huge amounts of calcium into shells daily, relying heavily on PTH and specialized bone reserves.
Wow. So wrapping this up, this journey through endocrine systems, it really shows how vital this internal communication is. From the simplest organisms to complex vertebrates, it's all about coordination.
Exactly. Whether it's a fast stress response or the slow, steady process of growth, or maintaining something like calcium balance second by second, hormones are the messengers making it happen, allowing animals to function, adapt, and survive.
It really is a testament to evolutionary ingenuity, isn't it? Taking ancient signaling molecules and refining them over millions of years to control just about everything.
And we're still learning. Unraveling the details of clock genes or the full range of actions of hormones like MSH, it just reinforces how interconnected and elegantly designed these systems are.
Just thinking about the sheer precision needed to keep your internal environment stable right now, minute by minute, it's mind-boggling.
And understanding it isn't just academic. It helps us appreciate life's complexity, and practically, it helps us tackle endocrine disorders in humans and animals.
Well, we hope this deep dive gave you some aha moments and a new appreciation for that incredible chemical symphony playing out inside all living things. What particular aspect of hormonal control stood out most to you? Thank you for joining us for this deep dive into animal physiology. We look forward to exploring more fascinating topics with you next time.